types of interactions between microbial populations. And in simplest type, we had two populations, A and B. We explained and we finished neutralism, which there were not any interaction between population A and population B. Right now, we are going to the second interaction with the name of commensalism. Commensalism. One is not affected or zero, but the second one gets benefited. It is positive, plus, positive or plus. Now we go through the commensalism. Commensalism. What is that? One population benefits and the other remains unaffected. Okay. Commensalism. Mensa is a Latin word with the meaning of food table. And com is related to the common. So common food table for two populations. But in such a way which one population in one side benefits and the other population on the other side of the table unaffected. We want to know how it is so. How it is so. First, I give you one example related to the animal. Then we go to many examples in microbial world. Suppose lion going to different places and as a predator find a deer, find cattle or sheep or anything and kill it and start to eat this prey. After some time, which lion is not hungry, going, but some meat remaining, and there are many followers for the lion. There are many followers for the lion, like fox, like wolves, and the other animals. And they start to eat the remaining meat. So why do they follow lion? Because after predation, because after lion catch a prey and eat it, going, and they can eat the remaining food. So they are benefited. Followers of the lion are benefited. Okay? But what's about lion? If they are there, lion going. If they are not there, lion going. In any case, when eat meat and when lion is not hungry, leave the place and going. So lion in these interactions with its followers unaffected. It is not important. No follower, one follower, 100 follower are there. For lion, there is no importance. Okay? So from this relationship, lion unaffected, but followers affected positively. They are benefited. Yes. Suppose I am sitting in my car and I have monitor. I'm watching football. Yes, national team, for example. It is not important for me. One person from outside look the monitor, 2% looking, 10% watching, or no person watch. I am watching television, so I am not affected from the presence other people around the car. But the other people are benefited. They like it. Okay? So they are benefited. I am in my home. I play violin. It is not important for me in the street, in neighbors, and other people here or not here. I'm doing for myself. But I'm doing violin very well. So the others benefited. They are happy to hear violin. Okay? So it is example not related to the microbes. Now, going some examples related to the microorganisms. So now you understand we are talking according interaction, not according eating or not eating. We are talking in the interactions between lion and followers. Lion benefited, harmed, or not affected. We say not affected. We ask followers of lion, affected, affected positively, negatively, or not affected. We say positively. So it is the meaning of commensalism. One population benefits and the other population remain unaffected. One example. Suppose you have one tube containing liquid medium, or a flask containing liquid medium. Shake it very well, and then inoculate anaerobic bacteria. What's happening? There is no growth because the tube or flask are full of oxygen. So anaerobic bacteria cannot grow. Now inoculate one facultative anaerobe. So facultative anaerobe can grow in the presence or in the absence of oxygen. There is indifference. They can grow in the presence or in the absence of oxygen. But if it is oxygen, use it. First choice is using oxygen for facultative. So what's happening? When we have one tube with two population, one is obligate anaerobe, cannot grow. One is facultative, which using oxygen. As much as facultative anaerobe using oxygen, oxygen is less and less and less, and situation goes better for anaerobic, obligate anaerobic. And in a time there is no oxygen at all, and anaerobic bacteria can grow also. So now what's happening? If obligate anaerobes present or obligate anaerobes absent, facultative use oxygen, so affected in this relationship. Facultative anaerobe and affected, 0. Because if the other group present or absent, they use oxygen. But what's about obligate anaerobe? They are benefited because the other group use the toxic substance for them. What was toxic? Oxygen was toxic for them. So facultative anaerobe doing detoxification, removing the oxygen. So in this example, facultative anaerobe and affected, but obligate anaerobe is benefited. So this relationship is commensalism. This relationship is commensalism. One population modified the habitat in such a way that another population benefits because the modified habitat is more suitable to its need. Population A, facultative anaerobe, utilize oxygen and lowers oxygen tension. So it is detoxification. With population B, a habitat, the new habitat is suitable for growth for them. So benefited. So population A unaffected or 0 in this interaction. Population B benefited or positively affected in this interaction. So here you see there is no talking regarding to eating any food. It is regarding to changing the environment. One microbe, population A, changing the environment in such a way which population B can live, can grow, can multiply. Another example is related to the production of growth factors. Forms the basis for many commensal relationships. In most microbes and most bacteria, they produce growth factors for themselves. Growth factors like vitamins, like amino acids for themselves, and some extra items excreted outside themselves, and the others can use them. So for example, population 1 produce vitamins and amino acids and excrete some amounts of them outside. If there are any microbes there, they do it. If there is no microbes there, they do it. So they are unaffected. But population 2, which use these vitamins and amino acids, benefited. So population 1 and affected, 0. Population 2 benefited or plus. One example, Flowobacterium brevis excrete a growth factor with the name of cysteine. If another population is there, or if there is no any another populations, Flavobacterium brevis produce cysteine. Okay, so unaffected. But the other populations like Legionella pneumophila use cysteine and benefited. So Flavobacterium brevis unaffected or zero, and Legionella pneumophila plus positive or benefited. So this relationship between Flavobacterium and Legionella pneumophila is commensalism. Commensalism. Note, production of growth factors and excretion allow fastidious microbial populations to grow. Production of growth factors. Now we are focusing, we are talking related to the fastidious bacteria. Fastidious bacteria. Suppose you have some children at home. One of them says, I don't like meat, I don't like fish, I don't like what, I don't like that. Yes? So this child is fastidious. But you have another child, he says, Mother, I like whatever you prepare. I like all of them. So this child is pretty well. And she is not fastidious. She is not fastidious. Accordingly, in the lab, so there are some fastidious bacteria which they cannot build, they cannot prepare some growth factors by simple materials. So along with nutrient agar, you prepare blood agar, chocolate agar, serum agar, enriched media. So why do you prepare enriched media? For growth of fastidious bacteria. But it is in the lab. What do they do when they are in the environment? Who prepare those things which they cannot produce? In the environment, fastidious bacteria are the followers of another microbes which can excrete those growth factors. They go in following another microbes which can produce those growth factors. So production of growth factors and excretion allows fastidious microbial populations to grow. So fastidious microbial population going, following the others, and by help of them they can grow. There are some examples. Sometimes changing of insoluble compounds to soluble ones. You know, bacteria don't have mouth like us. So they absorb nutrients by passive diffusion, active diffusion, active transport. Yes, they should make food in soluble. They cannot absorb insoluble substances. So some microbes doing solubilization for themselves, but the extra solubilized substrates can used with those microbes which are defective for solubilization. If population 2 is present or if population 2 is absent, solubilizers do the solubilization, so they are unaffected. But the followers get benefited because they can receive the soluble substrates and use them for their growth. Or here, for example, see desulfovibrio. If methanobacterium present or if methanobacterium absent, do this. Do what? Desulfovibrio use the sulfate and lactate as substrate and producing acetate and hydrogen. If another population is present or absent, no difference. So desulfovibrio in this interaction is unaffected by another populations. It is zero, but another population, methanobacterium, need these products coming from the desulfovibrio for changing carbon dioxide to methane. Okay? So methanobacterium has a good friend, the desulfovibrio, because receiving these substrates. But desulfovibrio doesn't need anything from methanobacterium, doesn't want methanobacterium to do anything. So desulfovibrio unaffected, but methanobacterium what? Benefited. Desulfovibrio 0 and methanobacterium plus. Methanobacterium plus. Suppose you are going by your car to one place. You see one person is in the rain. If the person coming to your car or doesn't come to your car, you are going to your way. Okay? You are doing you are going your direction. This person don't change your direction. So you allow the person come in your car, and in the middle of the way, the person going out. You don't get money. You don't ask him to do anything for you. So you are unaffected, but that person feeling well and benefited. Okay? And benefited. So this is unaffected. This is benefited. This is benefited. Another example, population make compound available to another population without actually transforming the particular compound, mostly in soil. For example, all living things need minerals. Minerals like phosphate, like potassium, like calcium, and the other, they are not organic. They are mineral. But in most places with less raining, mostly minerals are in insoluble form. They are insoluble. They are not soluble in the water. So one population, for use it, must solubilize it. even another populations are present or the other populations are not present. So it is this population not affected. Do it. That the other populations which don't have the power of solubilizations get benefited because using the solubilized mineral population, one population make a compound available to another population without actually transforming the particular compound, mostly in soil. For example, population 1 by acid production can release bond compounds and use it. And population 2, without having the ability of acid production, can using inaccessible compound. These compounds which they were inaccessible now by acid production now are accessible for population 1 and for population 2. Both of them can use it. Population 1 conversion of organic molecules to substrate. See, when we want to eat, for example, there is one plate in front of us with one pizza or with one meat, so we cannot eat immediately. We should cut it. We should make them a smaller size because they should be smaller than the size of our mouse. Okay? Again, we put a small part of meat or pizza in our mouth and then by teeth again cutting and cutting and cutting. Make them smaller. Then we send them to intestine, again cut it and cut it. Then digestion, absorption to blood. And this glucose, sucrose, and amino acids going to cells and are used. In bacteria are look like human. They start cutting outside the cell. For example, suppose one bacterium or one fungus like to eat cellulose. But cellulose, pectin, chitin, the others are in big size. They are polymer. It is not possible for bacteria eat directly cellulose. So they produce exoenzyme. They excrete exoenzymes. And exoenzymes cut and cut the cellulose outside the bacterium in a smaller size as substrates like glucose, then start absorbing by passive diffusion, by facilitated diffusion, by active transport, digestion, and receiving the glucose. Okay? Now suppose there are two populations, one in the left and the other in the right. One in the left and on the table is cellulose. Population 1 has cellulase and population 2 doesn't have cellulase. So population 2 never can use it because doesn't have cellulase. So population 1, in the absence or presence of another populations, produce exoenzyme encoding and changing cellulose to glucose and use it. And some remaining ones can be used by population 2 also, because population 2 has problem with polymer. That population 2 doesn't have problem with substrate of polymer as glucose. So population 1, conversion of organic molecule to substrates. Population 2, using substrate. Example, population 1, fungi, by extracellular enzymes convert polymeric compounds, cellulose, into simpler compounds like glucose. Population 2, lacking those enzymes, can utilize those complexes. Now, it is another example. Is it clear? Is it any question in general or from this example? Is it clear? Commensalism? Okay. So there is no question. Very good. As I told you, we are not focusing or eating or not eating. We are focusing in the interaction. Okay? In the interaction. Another example is removal or neutralization of toxic material. For example, H2S is toxic for many microbes. H2S. But sometimes one toxic substance for one population can be used for another's and not toxic to those ones. So if population 2 is present or is absent, population 1 doing oxidation of H2S, and it is detoxification. This toxic substance removed. So it is good for population 1 because use it. And it is good for population 2 because they are killed by H2S and now there is no H2S. So they can continue life. So population 2 follower of population 1, but population 1 no difference. Presence or absence of population, the other population, okay? Sometimes detoxification is by precipitation. Suppose there is water in an environment, there is water, and water contains some heavy metals, and those heavy metals are toxic for microbes. Population 1 in presence or absence of population 2, no difference. Population 1 can precipitate, can send the heavy metals to the sediment, to down water, and send them outside the water. So it is detoxification, like detoxification of mercury. If the other populations are present or absent, so this population not affected. But the other populations which are sensitive to heavy metal are benefited. Example 2, precipitation of heavy metals like mercury and detoxification by sulfate reducers. So sulfate reducers are present and doing detoxification and removing, for example, mercury. If the others present or absent, but the others as followers get benefited. Another types of detoxification is immobilization. What is the difference between precipitation and immobilization? Precipitation means extracellularly make the toxic substance to going to the sediment, to precipitate, going down, going out of water. That immobilizations, some microbes can absorb the matter which is toxic for many populations can be used by some. They absorb as substrate and use the toxic substance in their body. When one population absorbs toxic substances from water, so water is purified. The others are happy. After immobilization of a toxic substance by one population, the other populations are happy. Detoxify compounds by immobilization. For example, reduction of manganese by leptotrix, then growth of other populations is possible. If other populations are present or absent, no difference. Leptotrix immobilize manganese. But the others, as the followers of leptotrix, it is important because leptotrix cleans the environment and the others can live only in clean environment. They are killed in toxic environment. It was what we talked regarding to the commensalism with many examples regarding humans, animals, and especially regarding to the microbes. So in examination, we don't ask about lion, wolves, and human. I explain them because you come to notice regarding to the subject and then very easily understand the microbial examples. Now we go through the synergies. So today in this lecture we go through synergies, and by synergies we will finish today's lecture. Synergism or proto-cooperation. Synergism or proto-cooperation. What is that? Again, we will come back to the table, if you remember. In commensalism, one benefited, one not affected. But in synergies, both populations benefited. Both populations benefited. Okay? Sometimes you can see two students when come to the class sit beside each other, yes? Why in all semester, why for many years two students sit beside each other? So because it is positive interaction. Student number one feel well to be with student number two, and student number two feel well to be beside student number one. It has sometimes many reasons. You can see two students come with each other to university, then sit beside each other, then going to the canteen, then going home, and at home call each other and talk again. So it shows they feel well to be with each other. Now we want to go some examples regarding to the microbes. Okay. Before that, if these two students, for example, one of them to be sick and for 1 month not coming to the university, the others continue life. Yes. If one going to trip for 1 year, the others, or both of them continue life. So it is not obligatory. Okay? It is not obligatory. They are with each other, they feel well, but at night, one going to his home, the other going to his home. Yes? And they don't have any problem. Now we want to have some examples in this regards about microorganisms. When these positive relationships for both populations is related to the food, giving food and receiving food, we call it syntrophism. We call it syntrophism. Interactions of two or more populations that supply each other's nutritional needs. Nutritional needs. Again, another example, a student. Suppose one student knows very well advanced bacteriology, but has many problems in ecology. And his friend is very well in ecology, but has some problems in advanced bacteriology. So these two friends can teach themselves. Each can teach his friend and overcome two problems, removing the problems. So at the end of semester, both of them knows very well both courses. But problem shouldn't be in the same subject. For example, if two students, both of them are strong in bacteriology and both of them are weak in ecology, so how can they help each other? In bacteriology, they don't need help. And in ecology, both of them need help and cannot do helping each other. So when we are talking about synergies, there are two populations. Population A has problem A, and population B has problem B, and each population solve his friend problem, and they feel well to be with each other. They feel very well to live with each other. For example, E. coli cannot prepare ornithine from simple substrate. E. coli need receive ornithine from external source. But Streptococcus faecalis can produce ornithine from arginine. There are amino acids. So Streptococcus faecalis use arginine and change it to ornithine, which is needed by E. coli. So E. coli feel well to live with Streptococcus faecalis. And on the other hand, Streptococcus faecalis need putrescine. Which E. coli produce it. So Streptococcus faecalis feels well to live with E. coli. So E. coli has problem, Streptococcus faecalis has problem, but their problems are not the same. Their problems are different, and these two friends help each other to overcome to their disabilities, So when Streptococcus faecalis and E. coli live together in a tube, they feel they are lucky. They feel they have good friend. Okay? But it is not obligatory. If Streptococcus faecalis is not there, E. coli going and finding another friend. If E. coli is not there, Streptococcus faecalis going and finding another friend. But they need friend. Outside lab, in the lab, we prepare their media. We prepare enriched media. Okay, can you return it? You mean synergies from first? In synergies, there are two friends. Each has problem, but their problems are not same. And each help the other to overcome the problem. For example, E. coli need ornithine, cannot prepare it, cannot build it from simple substrate, and receive ornithine from Streptococcus faecalis. So E. coli feels has a good friend which give E. coli what needed. And on the other hand, Streptococcus faecalis need putrescine, which E. coli produce it and give it. So Streptococcus faecalis think has a good friend, which complete his disability. So E. coli give putrescine to Streptococcus faecalis, and Streptococcus faecalis give ornithine to E. coli. Both feels quite well. Both feel they are lucky to live with each other. Okay? But this friendship is not obligatory. If Streptococcus faecalis is not there, E. coli going and finding another microbe which with ability to production of ornithine. If E. coli is not there, Streptococcus faecalis going and finding another microbe which produce putrescine. Clear? Understand or repeat again? Dear student, Murtaza Sejil Jasa, did you understand? Is it needed to repeat again? Okay. Another example. Suppose there is factory for airplane. Most countries cannot build complete airplane. In one country, for example, France and USA. For example, the body and wings are produced in France and the motor is produced in USA. Then they come in combination. So both of them are happy. USA help France and France help USA. For what? For production the airplane. So they have problem, but their problems are not same. They have different problems and both help each other to complete themselves. For example, manufacture of car. Some countries preparing the body and another country prepare the engine. Then they come together and it change to the complete car. Sometimes you buy a computer. When you open the computer, you see it is completed with many parts. One part is from Taiwan, one part is from China, one part is from Japan, one part is from different countries. So sometimes it is combination. One cannot complete the product, so need help. Sometimes one microbe is not complete, cannot grow, need help. So E. coli need Streptococcus faecalis help E. coli, and now they can grow. In minimal medium, Streptococcus faecalis produce phenylalanine that requires folic acid. In opposite direction, Lactobacillus produce folic acid, which Streptococcus needed, but require phenylalanine, which Streptococcus faecalis produced. So Streptococcus faecalis and Lactobacillus are good friends. Streptococcus give phenylalanine to Lactobacillus. Lactobacillus give folic acid to Streptococcus faecalis. So they are good friends. They cover their problems. They cover their friends' problems. Together, the organisms grow quite well. Example 3. Nocardia metabolize cyclohexane as substrate and produce some products. These products are used by Pseudomonas. So Pseudomonas happy because receiving some products from Nocardia. And Pseudomonas produce biotin, which needed by Nocardia. So Nocardia and Pseudomonas, when they are living with each other, both of them are happy. Why? Because Nocardia and Pseudomonas have problem. Nocardia covered the problem of Pseudomonas, and Pseudomonas covered the problem of Nocardia. Okay? So it is now clear. If give one food and receive another food, and both population now can grow well, it is synergies or syntrophism. Synergies or syntrophism. Another example, some microbial photoautotrophs like light chlorobiome. You know, some groups of bacteria don't produce oxygen during photosynthesis. They use another minerals. Light chlorobiome produce formate and sulfur, which they are used by another populations with, like spirillum and the sulfur vibrio. So spirillum and the sulfur vibrio are happy, receive food from photosynthetic microbe. On the other hand, spirillum and the sulfur vibrio produce H2S, hydrogen sulfide, and carbon dioxide which both of them are used for this photoautotroph. So this one give food to the second population give food to first population. Colorobium and spirillum, both of them feeling quite well. They feel they are good friends and they are very helpful to each other. But there is not only exchange of food. One another thing can be observed. If you carefully look at this image, you can see when spirillum and the sulfovibrio produce H2S, hydrogen sulfide, hydrogen sulfide is toxic for them. So spirillum and the sulfovibrio during metabolism produce H2S, which has negative feedback on them. So when H2S is used by light colorobium, it is the toxification. These toxins for spirillum and the sulfovibrio are removed by colorobium. So in brief, spirillum give only food to colorobium, but colorobium give food and detoxification also. So colorobium is doing two jobs for spirillum, spirillum doing one job for colorobium. Spirillum only give food to colorobium. Colorobium give food and receive toxin and detoxify it. Note, conversion of hydrogen sulfide to elemental sulfur is the detoxifying step, as H2S concentrations kill spirillum and the sulfovibrio. Another example, again another photoautotrophs which produce oxygen and organic compounds. Light algae produce oxygen and organic compounds. So most bacteria like it. Most bacteria like organic compounds, like oxygen. Only obligate anaerobes don't like it. But facultative anaerobes and obligate aerobes all like it. So bacteria think algae are good friends. And bacteria use organic compounds and oxygen and produce carbon dioxide and vitamins. So carbon dioxide and vitamins are very nice for algae. So algae and bacteria are good friends. They give and receive foods. They give and receive foods to each other. Wherever there are any algae, many bacteria come there. And there is one definition with the name of chemotoxis. Who can give me a brief statement showing what is the meaning of chemotoxis? What is chemotoxis? What is chemotoxis? Who can write me in brief and very simple statement, simple sentence? What is chemotoxis? Okay, right now 18 students are present in the class. And I want to know who is the first student which write me the meaning of chemotaxis. Chemotaxis. Nobody knows it? Okay. Dear student, Murtaza Sajil Jasem is writing, and Ali Abdul Qadir Saadi. It is movement of an organism in response to a chemical stimulus. Okay, but it is half of the story. What is the remaining half of the story? The movement, the motion of an organism in response to a chemical stimulus. Chemical attraction is the movement of a living being or a specific entry in response to a chemical catalyst. Physical cells, bacteria, and other monocell, multi-cell organisms direct their movements according to some chemicals in their environment. Okay, the directed of cell in response of chemical stimulus. Stimulate. See, we have two types of stimuli. We have two types of stimuli. Some stimuli, some chemical substances are nutrients, and some the others are toxic substances. So, if there is nutrients in the environment, microbes going towards it. And if there is toxic substance, microbes go far from it. So it is chemotaxis. Okay? Chemotaxis is going towards a nutrient or escape from toxic substance. It is chemotaxis. Suppose when you are going in the street, most restaurants send the kitchen smelling to the street, because when you smell it, you try to go and buy it. Yes, it is according to chemotaxis. But if you go in dirty place and you smell bad things, you want to go far from it as soon as possible. So going through nutrients to get it, or going far from the toxic substance, try to get rid of toxic substance, it is chemotaxis. Okay, so why wherever algae are present there are many bacteria? Because bacteria, according to chemotaxis, going and living around it. Why? When there is the river, people are there, according to chemotaxis. Why? When there is a good place, people go there, according to chemotaxis. People, when there is a place for garbage, for bad things, for waste material, people go far. It is according chemotaxis. Okay. So wherever light algae are present, many bacteria go there and live beside them, and then they help each other, and both of them feel well according these interactions. Okay, the last example to finish this subject, and next week going to the mutualism. The last example, for example, all previous examples give food, receive food, give food, receive food, yes? Give food, receive food, give food, receive food, and all were syntrophism. All were syntrophism. Here, Pseudomonas changing orsinol to organic compound, and they are food for Brevibacterium and Curtobacterium. So Pseudomonas give food, but Brevibacterium and Curtobacterium don't give food. So how it is positive? Positive. It is positive for Brevibacterium and Curtobacterium. Why it is positive for Pseudomonas? Because these organic compounds are toxic for Pseudomonas. If there is no Brevibacterium and Curtobacterium, these compounds kill Pseudomonas. So when Curtobacterium and Brevibacterium are there, use it as food. So benefited, plus. And this toxic substance for Pseudomonas removed by them. So Pseudomonas is benefited. So in this example, one received food, the other received detoxification. Organic compounds are inhibitory feedback on Pseudomonas. So unlike the other examples, which giving, receiving food, here giving food, receiving detoxification. Giving food, and both get benefited. Okay? It is sufficient. So next week we go through mutualism. Mutualism completely is the same of symbiosis or protocooperation, because it is both populations benefit from relationship. But the only difference is that mutualism, living of these two populations together, is obligatory. If they go far from each other, both of them go to die. But the previous one, synergies or protocooperation, was not obligatory. So not obligatory regarding synergies, I explained. Next week, I explain you many examples which two organisms, two populations live together, help together, and cannot go far from each other. Their lives are dependent on their friends. Okay? If they are going far from each other, they go to die. It is what? Next week we will go through it, inshallah. If you have any question, please ask me. If not, we can finish our lecture today evening. Is it any question?